Tēnā koutou, tēnā koutou, tēnā koutou katoa. Good afternoon. Welcome to all of you. Welcome to our March 2023 All Hands Quarterly meeting. Great to have everyone who is here so far. This is a sort of a regular thing that we try and do to keep in touch with everyone on the project. And we were last together in person last October in Boise for our annual meeting. And so in between times, we try and do this. So really excited to have all of you here for our quarterly meeting. My name's Andy Kliske, the Idaho EPSCoR Project Director. I note we are recording the session, and that is to make it available on the GEM3 website for anyone who's unable to be on and listen to the meeting, and in particular listen to our lightning talk speakers who are coming up here in a few moments. We're sort of in that very busy period of the GEM3 Track 1 EPSCoR project, year 5. And today we're going to highlight the research, outreach, and other work being done by our doctoral students. The doctoral research is a really core part of any Track 1, and it is for GEM3. PhD research, doctoral studies are a huge undertaking in their own right, and to undertake doctoral research as part of a large interdisciplinary, statewide, cross-institution project is an additional undertaking. So I do want to acknowledge all of the work that all of our graduate students are doing, but particularly our doctoral students. PhD work is monumental, and it's a very timely investment of time and effort. And many, if not all of our doctoral students have been with the project from the beginning, if not early in its stage, and have seen the project, seen GEM3 develop. So I do want to recognize them. And so the core of today will be our lightning talks. We have 6 presenters. 2 of those will be presenting recorded messages, Tyler Breach and Jonathan Masingdale, because they are just finishing up their genomics training workshop with Smithsonian, so they're unable to be with us. And then we have 4 live speakers. So great to have those, and that's going to occupy the core part of the meeting. We have a couple of minutes towards the end, some updates on GEM3 aspects, and then a few very brief announcements that are really important given where we're at and with things coming up on the project over the next several months or so. So with that, I'm going to hand off to our first lightning talk speaker. They each have 8 minutes to speak. In terms of any questions you have, for efficiency, I recommend that once the speaker has finished, that you use the chat function to ask them questions and or email them. There's really not going to be the opportunity for us to go into a sort of oral set of questions. And for Tyler and Jonathan, in any case, given that there's a recording, you'll have to follow up with them by email if you have questions. So that's how we'll handle that. Please utilize the chat function as you're able to. So first off, we have Lucas Grothfurthner, University of Idaho, doctoral student working on population structure and hybridization under contemporary and future climates in a heteroploid foundational shrub species, Artemisia tridentata. Over to you, Lucas. Thank you.
Hello, thank you for the introduction. Yeah, I am Lucas. I'm currently, can you all see my screen well? Fantastic. Yeah, I'm a grad student here at the University of Idaho with Paul Hohenlohe and Lizette Waits, and I have been working on sagebrush in the last couple of years. And finally, we're able to show some actual results that are currently under revision in Frontiers in Plant Science. And before we dive right into into our project, I want to remind everyone that we've been working on big sagebrush, Artemisia tridentata, and that species is very interesting because we have 3 very distinct subspecies that are separated by ecology, by their chemistry, and by their cytology. So we know that some of them are diploid and some of them are tetraploid. And typically the distribution of those subspecies follows an environmental gradient. But to this day, there is no genomic evidence for how distinct those subspecies actually are and what happens when those subspecies co-occur. So we set out to test a couple of hypotheses, and we predict that the diploid subspecies are genomically distinct, and we predict that hybridization will be occurring, but it will mostly be limited on regions where the subspecies co-occur, which are, those regions are also called ecotones. And we predict that the hybridization will be high at the same ploidy level, and it will be lower between ploidy levels. And then as ploidy level, or the number of complete sets of chromosomes, plays a very important role in big sagebrush, we want to investigate that group a little bit further, and we predict that the tetraploids will be of allopolyploid origin, and maybe both of the diploid subspecies are contributing to its formation. And lastly, we wanted to look how they are currently distributed and how it will change under future climate scenarios. And so we predict that in the future, we will see a range contraction and we will see a subspecies decoupling, which also minimizes the possibility for hybridization under future climate scenarios. And what we're doing is we first constructed a climatic niche model for the Western United States, or generally for the big sagebrush region for each of the subspecies. And we then specifically focused on those regions where subspecies were predicted to co-occur. And those are here in this dark brown color. And we then went into the field, and at each of those predicted co-occurring sites, we sampled a transect from one subspecies. In the high elevation, it was always mountain big sagebrush, to either basin big sagebrush labeled here in green, or Wyoming big sagebrush labeled here in brown. And we considered all those plots that we sampled in between as potential hybrid plots to test how many hybrids there are actually there. And when we arrived at our plots, we selected 20 individuals at each of those plots, and we then analyzed those plants for ploidy to assess, ploidy distribution, and mapped that out across the landscape. And then we also used this information to prepare our sequencing library. And we performed a ddRAD sequencing protocol on a NovaSeq S4 lane, and we utilized Sven's great reference genome that he provided, which allowed us to then perform polyploid genotyping and perform population genetic analysis. And so what we were finding is very surprising and quite contrary to what we initially expected, because the two diploid subspecies are, as we expected, quite distinct, but we do not see a lot of hybridization. In fact, here on this principal coordinate analysis that we first constructed, we only find 4 individuals that appear to be right in between those diploid subspecies, and otherwise those subspecies are very well separated genetically. And then we also identify perhaps 2 different lineages in the tetraploid groups here marked with those filled symbols. So with that data set, we also performed a structure analysis where each bar, it's hard to see, but each bar corresponds to one individual that we sampled. And it very much corroborates the findings from this principal coordinate analysis. The diploid subspecies marked here in blue and green, are very well separated from each other with barely any sign of admixture in between them. And we do see two distinct tetraploid lineages, and then that heat map that just came up here also further corroborates those findings that we have very distinct gene pools, but this heat map allows us to assess relationships between each individual, and those are then clustered together. So it appears that we have one very distinct group corresponding to our mountain big sagebrush, one distinct group corresponding to our basin big sagebrush, which further shows population structure, which is in accordance with a scenario of isolation by distance. And then we also find two distinct tetraploid groups here, this one and the one in the corner. And what is very interesting to see is that it appears that the relationships between individuals of those different tetraploid groups differs between the groups, where one Wyomingensis 1 here appears to be more closely related to basin big sagebrush, and the second one, Wyomingensis 2, appears to be more closely related with mountain big sagebrush. So if we take our ploidy and subspecies information together and plot that out on the landscape, it differs a little bit to what we initially expected, because we actually don't find any hybrids. We only find 2% of our individuals to be hybrids, and all these others along the transect are actually very well separated. And what I want to highlight here in terms of ploid is that we didn't only find diploid and tetraploid individuals, but also we found triploid intermediates, which have been excluded from further analysis. And sorry, my computer is really slowly dying here. And now with those tetraploids, we were really interested in how those tetraploids formed. And we utilized allele frequencies to assess the mode of inheritance. And I don't really go into the details of how that method works, but basically what I want you to show is that there are two main different possibilities, which correspond to an autopolyploid origin, where all allele frequencies are present. All genotypes are present at an allele frequency of 0.5, in contrast to an allopolyploid origin, where all individuals share the duplex heterozygote at an allele frequency of 0.5. And so if we apply that to our two tetraploid lineages, we identify both of them to be autopolyploid, so they did not include a different species for their formation. And if you remember that heat map from earlier, I tested those two different lineages for their relationships with different subspecies, and it turns out that this Wyomingensis 1 lineage that we identified is more closely related to basin big sagebrush, where the second lineage, Wyomingensis 2, is more closely related to mountain big sagebrush. And those are really cool findings, because we identified two independent or two very likely independent origins, where each of those has a different parent contributing to its formation. So, and as I mentioned earlier, we also wanted to see how it changes under future climate scenarios. And sadly, our results are indicating that the sagebrush habitat in general will lose depending on the climate scenario. So this RCP 4.5 is corresponding to a more optimistic carbon dioxide emission pattern, while 8.5 is the higher emission pattern. And regardless, for both of these, sagebrush will likely lose quite a bit of its current range. So we predicted around 37% total loss under the better climate scenario, and around 56% loss of the sagebrush habitat under the worst climate scenario. And we also predicted that the range where all the subspecies co-occurs, again here labeled in dark brown, will be reduced quite a bit. So we argue it is very important to focus on those regions where subspecies co-occur to maintain genetic diversity, and perhaps find individuals which are suitable for future climates. So with that, I'm sorry if I ran a bit long, but I want to sum our work up by we identified the subspecies, particularly the diploids, to be genomically very distinct. And in contrast to our expectations, we found very little hybridization at the same ploidy level, particularly at the diploids. But we did find increased hybridization between diploids and tetraploids, and also between the different tetraploid lineages that we encountered. And this pattern has been observed in other plant species as well, and is called whole genome duplication mediated gene flow. And this may be describing what we're finding in sagebrush very well. And also in contrast to our expectations, we find an autopolyploid origin for the tetraploids, in contrast to the expected allopolyploid origin. And we were also able to confirm a range contraction in the future and subspecies decoupling, which will limit the potential for hybridization quite a bit. So with those first findings, we're able to now assess the effects of the genetic separation and distinctiveness on the phenotypes, for which we'll use volatile compounds and coumarin compounds, for example. And because the separation between subspecies is so clear, we also want to develop a GT-seq panel, which is basically taking all the genomic information we gathered and utilizing that for rapid subspecies identification, so that other researchers working with sagebrush can quickly identify those subspecies. And lastly, that's because I highlighted that we also found those triploids, and there are not many systems where that so-called triploid bridge has been reported. So we're also able to assess the role of the triploid bridge for sagebrush based on at least 2 individuals that we found. And with that, I thank you very much for your attention. I also want to thank the labs that I've been working in, all my lab mates. I also want to thank Tom Parchment and Ovidio Pound for helping with the genomic analysis, and of course, Tammy and Rick for all their amazing help throughout those last couple of years for really enabling our research.
Great, thank you so much, Lucas. Wonderful.
Sorry if
carry on.
Longer.
No problem at all. So if anyone has questions for Lucas, please use the chat function to ask them those questions. Our next lightning talk presenter is Tyler Breach from Idaho State University, who is talking on intra-specific variation of rainbow trout in Western North America. And this is a recorded presentation.
Hi, everyone. I'm Tyler Breach, a PhD student at Idaho State University, and I am looking at the intraspecific variation of rainbow trout in Western North America. So rainbow trout are a salmonid species that have the largest latitudinal range of the salmonid family. As you can see in this map on the right, their native range includes most of Western North America, from Mexico to Alaska, and also extends to the Kamchatkan Peninsula in Russia, which isn't pictured here. The habitat complexity contained within the distribution, including different climates and recently deglaciated areas, has contributed to vast amounts of diversity within the species. This diversity has been recognized for many decades, but attempts to organize it have been confounded by local adaptations, phenotypic plasticity, and morphological overlap. So this is a map that I created with commonly accepted subspecies represented by different colored polygons, and additional hypothesized unique populations are listed underneath the corresponding subspecific area. Much of this map and rainbow trout diversity classifications in general are centered around phenotypic traits and major biogeographic features, such as watershed boundaries and mountain ranges. However, you can note the disparity in the size of ranges for several subspecies and the many distinct additional populations for several of these subspecies. Between 9 and 15 subspecies have commonly been proposed, depending on how lineages are defined. And this disagreement between identification of subspecies highlights the ongoing problem of classifying rainbow trout evolutionary lineages. Currently, as I mentioned, many of the suggested rainbow trout subspecies are based on phenotypic differences. So here are 5 examples of commonly accepted subspecies of rainbow trout native to the Western United States, generally classified by qualitative, morphological, and meristic data. While these 5 subspecies are generally accepted as subspecies based on phenotypic differences, more recent genetic research has revealed fine-scale population structuring and questioned the accuracy of using solely traditional phenotypic observations to define evolutionary lineages. So to date, no range-wide comparisons of rainbow trout genetic diversity have been conducted, but at a smaller scale, genetic data has shown results inconsistent with phenotypic classifications. For example, on the left is a map of British Columbia with 2 different clades represented by black and gray circles. Generally accepted subspecies for the area were coastal rainbow trout in the west and red band trout in the east. However, McCusker et al. Did not find this east-west division, but rather clades were more intermixed. On the lower right is a phylogeny of rainbow trout from a more southern region of their range, specifically the Oregon Desert basins and the surrounding watersheds. Before this study, the rainbow trout in this area were considered one population of a subspecies. Kearns et al. Found 3 different lineages seen on the right, written on the right based on genetic data. Whereas McCusker et al. Found more of a intermixing of clades. However, the study by Kearns et al. Showed a pattern of sorting of lineages by watershed. As clearly as phenotypic classifications are uncertain, so are genetic classifications and overall organizing factors of rainbow trout diversity. So as rainbow trout are becoming of increased conservation concern in their native distribution due to environmental change and population shrinkages, identifying lineages, understanding the evolutionary history, and mapping patterns of divergence is necessary for assessing population viability and adaptive capacity for future conditions. So the research objectives for my current study, I should say, are to identify and map major evolutionary lineages of rainbow trout in North America, and then to look for concordance between genetic lineages and morphometric variation. So to collect samples for this, we collect 30 to 35 fish from each population, which we then take a fin clip from and high resolution photos for morphological comparisons at a later date. The green dots on this map indicate where I currently have samples for either the genetic or the morphometric data set. So the current estimates that I have for populations for both are about 170 populations for the genetic data and about 115 populations for morphometric data. This year, I am planning to sample throughout California and to sample in Oregon, where there are no representative populations currently sampled. Additionally, I'm working with collaborators, agencies, and other researchers to obtain samples so that I can include populations from Alaska, British Columbia, and Washington. In the future, I'll be looking for genomic structural variants among rainbow trout populations. So here's the map one more time to show not only the differences in subspecies proposed, again shown by different colors, but also this geographically broad range of rainbow trout. As I mentioned before, within this range, both between and within putative subspecies is a vast amount of diversity, including an array of life history strategies and different ecotypes. So I will sample select populations to examine genomic structural variants such as inversions or copy number variants to see if they correspond with different traits of these populations. And I would just like to acknowledge everyone that has helped me so far, and there are many others that have played a key part already. With that, if you have any questions, feel free to reach out.
Great. So, we do thank Tyler Breach in absentia for his great talk there, and please email him if you do have any questions for Tyler about his presentation. And then we next have another in-person presenter. I'm delighted to introduce Hayley Netherton Morrison from Boise State University, who will be presenting on her work on linking values, so social values, to management attitudes in the changing sagebrush steppe. Thanks, Hayley.
Sorry, I had to refine my Zoom controls to unmute myself. Okay, so in working landscapes undergoing social and ecological changes, it becomes increasingly important to understand the values people hold and their implications for the support of management strategies. And because we want to be able to determine who holds what values and where they live on the landscape, and that this could help prioritize management actions, we wanted to answer two questions. The first of which being, how do values for sagebrush steppe landscapes vary? And the second being, how do attitudes towards sagebrush steppe management vary spatially? And so to answer these questions, we used a statewide phone and web-based survey measuring opinions on sagebrush landscapes, rangelands, and public lands in the state. We had 1,048 respondents, and they were a good match to the population of the state. We had a few lower population counties that didn't have any responses, and we also purposefully oversampled based on stakeholder interests in 2 rural counties of Idaho. Today, I'm going to focus on 4 questions from the survey. The first being the general importance of sagebrush landscapes. The second being values for sagebrush landscapes, which was a set of 15 values that we asked about. Next, the preferred action to manage sagebrush landscapes in general. And then finally, attitudes toward different actions to manage recreation in these landscapes. And so first, I'll share results comparing public values and attitudes based on the personal importance of sagebrush. And then I'll share some of our modeling and mapping work to develop county-level estimates of management opinion and conflict using multi-level regression and post-stratification and the potential for conflict index. And so starting out with the values piece, we formed 4 groups of respondents based on the level of personal importance for sagebrush steppe. And so folks were asked to respond to the question, how important is sagebrush to you personally? 1 being not at all important, 4 being very important. And we're going to be comparing those values and recreation management attitudes based on these groups that we've put them in. And so the next slide will show a series of spider plots with the 15 values for sagebrush landscapes around them. And how to interpret these is that inner loop is a 1, meaning that the group the median rating was not at all important, and a 4 being very important. And so each individual value will map onto the spider plot such as this. And so what we're really seeing when we're comparing these groups is that even in cases where sagebrush steppe isn't personally important, we still see that these broader benefits types of values such as wildlife, ranching, recreation, and the iconic nature of the landscape are valued. And as personal importance increases, so does the number of these more personal connection type values for the landscape, such as spending time with family and friends, the feeling of home, and spiritual connections to the landscape. Next, we wanted to understand the relationship between these groups and management attitudes for recreation. And so we asked about the acceptability of 5 different actions to manage recreation in Idaho's sagebrush landscapes. The first being establishing new trails, then setting and enforcing rules that limit recreation access, then providing financial support for managing areas affected by recreation, bringing people together to make decisions about recreation issues, and doing nothing. And so today I'm just going to focus on the 2 extremes. And so for each of those actions, they were asked to rate strongly agree to strongly disagree. And so we're just going to focus on those 2. And so here we see that collaboration was the most strongly accepted action across the groups, and do nothing was the most strongly opposed. And there is a general trend toward higher rates of strongly agree as personal importance of sagebrush landscapes increases. We also found that the group, the not at all important group, more frequently selected the I don't know option, showing some uncertainty in their opinion on how to manage recreation in these landscapes. And so in addition to this recreation specific question, we also discussed, we also asked respondents to select the most acceptable action for ensuring sustainable sagebrush landscapes. And so for the remainder of this presentation, I'm going to be focusing on those county level estimates for management opinions that I mentioned previously. And so looking at this general question of what is the most acceptable way to ensure sustainable sagebrush landscapes, for those same 5 actions that we asked about in the recreation context, we see that for the most part, the enforce and establish actions are viewed as the most acceptable. We also see that enforce was the most frequently selected action across most counties, and particularly in the northern Idaho counties. Additionally, what we wanted to highlight was this finding that Owyhee County has a very high acceptance rate for collaboration. And this might not be too surprising, depending on your familiarity with Owyhee County. This is home to one of our GEM3 stakeholder advisory groups, but it also has a really strong history of collaboration through things like the Owyhee Initiative and one of the more active sage grouse working groups across the state. And so switching back to our focus on recreation, we see that all the actions except for do nothing are viewed as acceptable across all the counties, with collaboration having the highest acceptance rates. And so for the next couple of figures, I'm going to go back to focusing on this enforce action, since it's strongly accepted for both ensuring the general sustainability of sagebrush steppe and for managing recreation. And so this next map is going to be a bivariate map comparing the county level estimates for enforcement as the most acceptable action in the general question, and that's going to be on that orange axis. And then comparing those with estimates for agreement that enforcement is acceptable to manage recreation. And so that's along the blue there. And so we see that enforcement is acceptable, but not necessarily the preferred action in many of the counties. And so notably, we're seeing the strongest support for enforcement in northern Idaho, where there's less sagebrush cover, meaning enforcement isn't necessarily happening locally, but it's still supported. And then going back to using Owyhee County as an example, our stakeholder group has really expressed the concern around enforcement of recreation rules. It's a huge problem trying to enforce recreation rules right now in that space. And although the enforcement is acceptable, as we can see here, it's not necessarily preferred to other types of actions to manage recreation, which is showing that there may be some on-the-ground realities that are driving that difference in opinion from other counties. We also wanted to understand the conflict over enforcement to manage recreation. And so we calculated potential for conflict index scores for each county. And so those are going to range from 0, being no conflict, full consensus, to 1 being high conflict, low consensus. And across the state, we actually find moderate conflict potential for enforcement. And of the 5 actions that we asked about, enforcement was among the most polarizing. And in that there was this higher level of disagreement, this moderate conflict potential. And so why might we be seeing some of these patterns in enforcement opinion? And so an additional question that we asked as part of a larger set of questions about recreation was To what extent do you feel your recreational use of public lands has negative or positive impacts on the environment and other public lands users? And what you might be seeing is that people largely don't view themselves as part of the problem. They primarily view their impacts as being positive or neutral. And so since very few people believe that they're part of that problem, but they still believe that there are multiple problems related to recreation, there may be this mismatch between what the perceptions of impacts are and how people feel it's appropriate to actually address them. And so in summary, some key takeaways from our work so far is that Idahoans really do value sagebrush landscapes in diverse ways. The more personally important sagebrush step is, the more strongly Idahoans agree that we need to take action on recreation management. And we're also seeing that it's really complex trying to balance sagebrush conservation and management because not only is the landscape valued, but so is recreation. And so some of the actions that we asked about can be very polarizing in opinion. And we're also seeing that differences in that acceptance of the different management actions may be linked to the different values that people have, as well as their perceptions of things like their own personal impact on the landscape. And so this is my email in case you have questions, and I'm happy to answer anything via chat. Thank you.
Thank you so much, Hayley. That was a great presentation. Please use the chat function to ask Hayley questions and or email, but utilize the chat function. As our next speaker, presenter Jonathan Massingale from University of Idaho. This will be a recorded presentation. And Jonathan's lightning talk is titled Red Band Trout Common Garden Overview, Status and Results.
Hi, everybody. My name is John Massingale, and I'm a PhD student at the University of Idaho in the labs of Drs. Brian Small and Chris Cadill. And thanks for joining me on an update of our red band trout common garden experiment. So first, I'm just going to review the common garden purpose and workflow as part of the trout mechanisms section of the GEM3 project. First, our big research question is trying to answer if interpopulation differences in plasticity contribute to adaptive capacity. And to get at this, we collected wild red band trout fry from streams throughout Idaho across an environmental gradient ranging from cold mountain streams to desert streams that get significantly warmer during the summer. We then reared them at 3 temperature treatments ranging from optimum to stressful and conducted experiments to understand how both genetics and environment play a role in temperature tolerance and performance. And we then aimed to quantify physiological and behavioral traits and conduct genomic analyses to discover genetic mechanisms for these traits. Our results will then inform modeling efforts to predict population persistence across space and time, and an overarching goal of using red band trout in the mechanisms portion of GEM3 is to create a model species, providing a basis for future research. So next, I'll be briefly going over some of the results found from the first cohort of fish that we reared in the common garden. So here are a few slides taken directly from Dr. Jiangxi Chen. And this shows some of what was found. And first, he conducted an experiment called critical thermal maximum, or CTmax, which measures the upper thermal tolerance of an individual. Now, in this experiment, fish are subjected to an acute warming treatment, whereby water temperature is increased at a specific rate until fish are unable to maintain an upright position. And his results showed that little variation in thermal tolerance was explained by genetic variation between populations, but that acclimation temperature played a large effect. Now, the functional level, measuring maximum heart rate in a similar experiment to CTmax, where fish experience warming with an ECG monitor, he found differences between ecotypes, but mainly at the lower acclimation temperatures and not at the higher stressful acclimation temperature. He also measured swimming performance, in which he discovered no acclimation effect for desert or cool montane fish, or between the 2 lower acclimation temperatures, but found that montane fish swimming performance decreased significantly in the 21-degree acclimation treatment. And finally, we measured growth performance over the course of about a year, and we found that body size was similar across all populations in optimal acclimation temperature, but that cold montane fish actually were larger at the final measurement than cool montane and desert fish. Now, overall, the stressful acclimation temperature resulted in lower growth rate among all ecotypes. So to summarize, these results imply that most montane red band trout will probably have the capacity to tolerate temperatures higher than their current habitat extremes using phenotypic plasticity. And desert fish are approaching their hard ceiling for upper thermal limits and are expected to rely on thermal refugia during hot summer days rather than plasticity. And as for performance, desert fish likely have expanded their thermal performance close to their upper tolerance limits. And under predicted global warming scenarios, cold montane fish are expected to adjust performance traits such as cardiac response with a combination of genetic by environmental interactions. So that was the first cohort. With our current cohort, we've added a diel fluctuating acclimation treatment paired to each constant temperature treatment, which fluctuates 2 degrees C above and below each constant treatment during a given daily cycle. So for example, we have the 21-degree constant temperature up here, and the paired diel periodicity would fluctuate between 19 and 23 degrees C daily. And the idea behind this is to replicate a more natural regime that you might find in a stream during summer. Traditional common garden experiments generally use constant temperatures, but studying the effects of fluctuating temperatures could be more ecologically relevant and help elucidate the effects of experiencing both a higher brief peak temperature and a lower recovery period every day. So next, I'll just cover what's happening with my current work in the common garden. I've also collected cardiac performance data using the same methods as cohort 1, but I'll be adding the comparison for the results of the constant versus diel fluctuating acclimation treatments. I'm also currently in the process of collecting my own set of CT max data. Now, for cardiac performance, I'm focusing on a performance metric called Arrhenius breakpoint temperature. This is when the rate of change decreases, which you can see in these breaks on these plotted lines. This is used for a good proxy for understanding the thermal range of an individual's performance more than just the upper thermal tolerance. So we could think of this as the range of fish could efficiently digest food, grow, mature, etc., rather than just survive in. And as I'm interested in habitat selection, this is a good metric to compare with my behavioral studies, which I'll cover now. So for behavior, I'm primarily looking at temperature preference, and I'm doing this using an apparatus called a shuttle box, where a fish can move between 2 arenas which maintain a 2-degree difference at all times. And temperature in the system changes depending on which arena the fish occupies. So for instance, if a fish occupies the arena on the left here, that's in blue, the system temperature will decrease, while if it occupies the one on the right, the system temperature increases. Equilibrium is considered to be achieved when a fish spends equal amounts of time in both arenas, and the end result of the experiment is the rolling median of occupied temperatures after a 12-hour time period. And I think this will pair nicely with my heart rate data, and I expect that temperature preference will fall within those Arrhenius breakpoint temperature results. Following these experiments, I'll analyze these data to quantify the phenotypes and conduct a genome-wide association study to discover candidate genes associated with these traits and determine genetic differences in these traits among populations. I also plan to conduct experiments and collect RNA tissues for RNA sequencing to understand the influence of environment on gene expression. And I'd also like to note that we're currently waiting on RNA sequencing results for cohort 1. We hope to receive those data soon for analysis. So that's the update on the red band trout common garden, and thank you very much for watching.
Great, and again, thank you to Jonathan and Abstensia for his presentation. Please direct any questions you have for Jonathan via his email. And so I'm now delighted to introduce Andrei Zayats from Boise State University, whose live presentation will be on scale-dependent interaction between structural heterogeneity, wildlife effects, and recovery in arid shrublands. Thank you, Andrei.
Thank you. Okay, can you all see my screen and hear me? Yeah, great, thanks. All right, so we all know that ecosystem structure is incredibly important for the maintenance of biodiversity healthy animal and plant communities across the globe. In today's presentation, I want to show you some exciting updates from our lab that show that the relationship between ecosystem structure, and specifically structural heterogeneity, and processes like related to sagebrush community resilience could be scale dependent. So we can think of structure as the distribution of plants in 3D space and the variability of that distribution, whereas a function is some biological process that takes place within an ecosystem and could include photosynthesis, recovery, resilience, etc. Within those categories, we can also think about structure and function at different levels. So for example, on the structure side, we can think of the distribution of leaves in space, whereas on the functional side, the distribution of those leaves will determine for example, physiological processes or photosynthesis, depending whether the leaf is located on the top of the canopy or inside of the canopy. And so interestingly, the technology that has allowed us to map vegetation in 3D space very precisely, like drone technology or other sensors, collect information about structure and function at multiple levels, from individual organs, individuals, populations, and communities. So here's what I mean on an example of the sagebrush step. So generally, we can think of sagebrush, healthy sagebrush ecosystem characterized by sparse vegetation with well-defined interspaces between the plants. And so when we look at this scene, especially the unburned part of this landscape, the first pattern that our eye catches is the distribution of adult plants. But as this drone flies over and we get a look at what happens in the interspaces of the shrubs, we can see that those interspaces could be filled with shrub recruits or other vegetation, and the interactions between those plants in the interspaces may be happening at a different scale. So how do we quantify those patterns at different scales? One way to do that is to consider structural heterogeneity coming from the drone image, like this canopy height model in the bottom. The black line here indicates the fire line. On the left, we have the landscape that didn't burn. On the right, the landscape that burned. And so instead of summarizing structural heterogeneity as for the entire site, we can decompose this metric into distinct scales. So heterogeneity that happens at the scale of 4 centimeters, 8 centimeters, etc. And so now with that in mind, we can answer how does structural heterogeneity changes in response to wildfire? And also, what is the relationship between structural heterogeneity and recovery? And so keeping in mind that we now have more than one way to quantify heterogeneity. So to answer those questions, we collected data from 10 sites in southwestern Idaho, and that followed this design along the fire line. And we distributed randomly the circular plots and mapped exhaustively all shrub recruits within those plots. And so to answer question 1, we're just comparing structural heterogeneity between burned and unburned sections of the plots. And to answer question 2, we're quantifying the relationship between recruit abundance inside the circular plots and the structural heterogeneity within that plot. So when we look at how structural heterogeneity changes in response to wildfire, it's predictably generally decreases. And here's how it changes across all of our sites that we sampled. And generally, we see that it declines, but also we note that this decline happens at a different rate. So some structural heterogeneity metrics were less sensitive to wildfire than the others. And we asked whether there's an agreement between which scale of heterogeneity is most sensitive. And we surprisingly find that the most sensitive scale of heterogeneity is actually happening, was quantified at an intermediate resolution, like from meter to 2.5 meters, and not at the finest resolution that we collected from the canopy height model that we collected with a drone. And so the sensitivity of this heterogeneity at the intermediate scale could be related to the presence or absence of adult plants. In contrast, when we looked at the relationship between recruits and heterogeneity, we found that the process of recruitment is likely happening across multiple scales. So we found that some of the relationships were negative and some were positive. And we hypothesized that the negative effects could be related to processes like competition, whereas the positive effects could be could suggest that we use structural heterogeneity as a predictive metric to quantify recruitment in post-fire landscapes. So when we looked at a closer look of the relationship between heterogeneity, recruitment, and the number of adult plants within the plots, we found that both the effect of adult plants and heterogeneity were positive, but the interaction between was negative, which leads us to a hypothesis that under low density or small competitive pressure from adult plants, the effect of heterogeneity is positive and is likely to improve recruitment, versus if as the number of adult plants increases, density dependence prevails, and the effect of heterogeneity becomes negative. So in summary, I want to just to say that scale decomposition of structural patterns gives us a more nuanced view of structure and functional relationships, like the wildfire effects or the post-fire recovery. And so with that in mind, we can use these metrics to better understand the drivers of resilience, the relationship between density dependence and structural heterogeneity, and also the role of density dependence in the recovery process. And as a second summary point, I want to highlight the work of an undergraduate student, Valerie Marie, in our lab, that has done a tremendous work helping putting this data set together and publishing it as data sets available to anyone, including the reproducibility workflows and the workflows that we use to process the raw imagery and the analysis. And so just want to give thanks to Andrew Child for his help in that process, and Donna Delpart for guidance as well. And so with that, I'd like to thank my co-authors and everyone in the Coghlan lab for helping with data collection and analysis and basically daily support. And with that, thank you.
Great, thank you so much, Andre. Great to see your work. Excellent presentation, thank you so much. If anyone that has questions, Andre, please use the chat function to ask any questions. We are going to just have a slight change in schedule here. Unless Spencer is on, if you can hear me. Our next speaker is Spencer Root, but I think Spencer is connected. So we'll continue with some of the updates and then return once we are able to connect with Spencer. So I'm going to start some key updates for all of us on the project. First one relates to annual reporting, and there is a change coming from NSF that will partly affect us. So we will continue to use the Pi-Star system that we've used each year of GEM3, including last year, that many of you are familiar with. That will be used to collect the information from each of you. The system is scheduled to open April 17, and with reports and reporting due by May 31 at the very latest for that. Second, we will not include, or we're proposing not to include, as part of that reporting, the AOP module, which is the one that's used for narrative reporting. And in liaison with the co-PIs and the leadership team, we will come up with a different approach, hopefully a more efficient approach in using the AOP. So you will not see the AOP module for those of you who have been involved in that piece of it. But thirdly, and importantly, NSF has created a new online reporting system for RII projects, track 1s and track 2s. The rollout for that has been slower than they anticipated. This new system is called eDocs, E-D-O-C-S, eDocs. And whilst previously it was going to be in place for April, that's been pushed back till July. So, which means we won't be using it for submission of our Year 5 annual report. But NSF has said that they will roll it out after that and will be requiring us to report via that. We don't know what that means and the extent to which there might be duplication of information that we're asking for you for the annual report that has to be submitted by the end of June, versus what will be in that eDoc. So just kind of a heads up, we will be monitoring that. We will try our best to minimize duplication of effort, but it's possible that some will be unavoidable because we don't know what NSF has got in and is requiring as part of the eDoc, and we obviously cannot wait until July for that to be able to report by June 30th. So that's the first one. We'll have more information about the annual reporting, and we'll be talking with each of your campus leadership teams about that to make sure that we understand that very clearly. The other item I want to report, which is actually listed at the end of the agenda, is on a no-cost extension for GEM3. We did submit a request to NSF just last week, and our program officer has responded very quickly to approve that no-cost request. That is at this point a 6-month extension request, and that will at minimum allow us to fund activity that's deemed a priority through the end of this fall semester. We're still figuring out, we've only just gotten the approval beginning of this week, and so we still have to work through and figure out what that means. But the short of it is that we have some of an extension, and we've been told that it is possible, depending on circumstances, we could apply for a further one. If we were to do that, and assuming we had funds remaining, that might allow us to support activity through spring of 2024. But that's really only a maybe possibility at this stage. In the request, the justification, one of the priorities we asked for the no-cost extension for was to support doctoral research. And so, we've just had a great set of presentations from 6 of our doctoral candidates. That work really is the core of the work being done in GEM3, and I do want to acknowledge and thank all of our speakers for their work. I mean, it's high caliber work. It's core GEM3 work. I also want to recognize the work done by each of their supervisors in supporting and encouraging and enabling all of that work. So that's certainly a priority that the leadership team, each of your lead and co-leads at your universities, will be looking at as over the next several weeks we try and figure out how we spin up this no-cost extension. So that's kind of hot off the press. We do have a few minutes available, so because it's quite an important thing, I'm happy to entertain any questions that anyone has about the no-cost extension, to the extent that it's possible to answer it at this stage. I do encourage you, if you do have questions, to talk with your, if you're at BSU, talk with Jan, talk with Peggy. And actually, on that point, I would note that Peggy Martinez has stepped up to take on the program coordination at BSU as we sort of farewell the role that Denise Pfeiffer has had as Denise moves on to another role at BSU. And thank you, Denise, for the role you've had. Then at ISU, please connect with Colden, Maury, and Kitty if you have questions. And then at University of Idaho to Chris Borlaug and Paul Townley. And you'll see more there. Okay, well, let's now move to the next update. Jen, if you'd like to update us on the workshop, your workshop building bridges. Thank you, Jen.
Yeah, thank you. Welcome, everybody. I think I've got an exciting opportunity. I'm going to share my screen here and show you a few slides. So, let's see, come on. All right, so some of you have heard about a bombardment maybe of TIP funding, so Technology Innovation Partnerships funding, and you may be use-inspired curious, or you might have heard about this TIP and wondered, is it for me? And so if you're curious or wonder if it's for you and how you might tap into some of that funding, I invite you to come to a workshop. So this is TIP's investment in this, and I'll say that, let me get rid of that. What you might see, what it might appear, and this is true, that most of the funding coming through TIP seems to be targeting private industry, and they were sort of their first targets. But they're thinking about expanding this, and in particular, the biology directorate is very interested in how biology side of NSF doing basic science might also be part of this new TIP environment. And so JD Swanson, who is our track 1 program officer, partnered me with Michael at the University of Hawaii, Manoa. We both have track, we're PIs on track 2 EP scores related to rules of life, much like our track 1 is. And we then put together a workshop, the first of hopefully many, not that we would lead, but that others might lead in other regions and other topics, about building bridges between use-inspired research and science-informed policy. And so the goal of this is sort of, anybody that's familiar with the biomedical side of things, this is translational ecology, or the valley of death between, bench side to bed is what they call it in biomedical, and we might call it from the lab to the landscape. And so how do you translate the basic science that we do from NSF into having benefits for society? And that requires this co-investment between public funds in our universities and the private funds that are out in the sort of economic environment that exists. And I'd say that, so there's a couple of things that NSF is doing. They're thinking about who these partners are. And so this is NSF partnering with other federal, state agencies. And then also thinking about not just private funds being the sort of sustainer of the products that we build, whether it's workforce or technology, but our local communities that might use those. And I think our GEM3 community is well poised to do this. So although private was sort of the first target, government, federal in particular, is going to be represented at this workshop. And this is sort of the idea, right, that although many of us might have relationships and partnerships with government, some of us might have relationships with private. What NSF wants is all 3 of these arms to be actually working together to drive technology and to identify these science-informed practices that we can do, in particular associated with climate change. The other outcome is just to diversify the careers that our students get. So instead of us all driving our students to academic careers, how do we diversify the skills and technologies and training that our students get so that they're better prepared for careers in both industry and government? Here's the QR code if you want to take it. And I'll also plug in, let me go back to the chat. Here is the website, so you can go and register. Again, this is funded between TIP, Bio, and EPSCoR has funded this workshop. Few other things. Think of this as the intentional co-development of the broader impacts of our proposal. So all of us have been doing, or at least thinking about use-inspired research and education. It tends to be the thing that we put in our broader impacts, not always, but sometimes. And think of this as having those conversations like what Haley showed us with our partners from the start to drive and inform the way in which we do our science and the questions we ask and where we ask those questions. There will be 2 virtual, all welcome micro labs. So this is the first, is Friday the 14th in April. April 14th from noon to 2 p.m. The second will be May 12th from noon to 2 p.m. And those are open for anybody that registers. You can come to those. We encourage people coming to both of them. And then based on the registered individuals, even if you don't come to the virtual ones, you can still apply, and we're going to have that application available by the April 14th deadline that you can apply for in person. For 120 people will come to Boise to actually map out things. I've got actually what I wanted to also show you then is just who's coming. So this is our data board. So we already have 500 applicants that have applied, which is awesome. We thought maybe we would get a couple hundred. And then here's the distribution. So, you know, over 300 are from academia, but we do have some government. So those of you that work with government partners, if you want to be prepared for calls that are going to come out of this use-inspired space, please have them come so they can be part of those conversations. You'll see that we want all our graduate students to apply to this. So we only have 42 graduate students, we have 38 postdocs, and we have 69 early career. So at your institutions, anybody, it doesn't have to just be GEM3 people, please encourage your graduate programs, your postdocs, and then early career faculty that your institutions have hired to sign up. I want to show you that Idaho's winning. So we have the most registered individuals across all the states. You'll see that every state and jurisdiction is represented here. And so let's get lots of Idahoans there. And so please tell your partners and our other EPSCoR jurisdictions. And that most of the folks coming, their primary interest is biology, which is great because bio funded this. And so they like to see this, but you'll see that there's quite a few engineers, quite a few, TIP related individuals. And then the last part I want to show is that the sort of alignment, we ask people to when they register, to where do you think your highest alignment with science-informed practices are? And, biotech is high, but also education. So clearly there's going to be a big interest in thinking about how we might change our curricula, change how we train our students. I think GEM3 is well positioned to lead in this front with our VIPs and our experiential learning and our connections with our predominantly undergraduate institutions. And that I'm going to stop there. I can give you examples, but I thought I'll stop there. And if you have questions about the workshop, I'll put back up real quick. Where did it go? I'll put back up the scan. So if you want to scan it again, and again, the website's there, and we've sent this out through our EPSCoR community, but if you want it again, let me know, but I'll give you just a moment to take that on, and then I'd be happy to answer or try to answer any questions you have related to the workshop.
Great, thank you so much, Jen. Thank you so much for spearheading this. This is really innovative and great to see. Are there any immediate or burning questions for Jen?
This is Sanford. I just noticed the 0 in registrants in crops and crop management. So seems like maybe this isn't getting out to a certain community of scientists. I'll do my best, but
yeah, so it might have been that we had an ag related one. And so it might have been that all those people, so Mike is very strong in the crop, agricultural, biotech side of things. And so I think when he looked through the list, he assumed that most people had actually, it might have been that we didn't allow multiple. And so people might have put themselves in the agricultural section of that. Let me just, I don't actually know where that went. Yeah, so there's 114 through the agricultural research, and then there was separate crops and crop improvement. And then the other was, it might have been that there was agricultural research and biotechnology were the two that people might have aligned more with. But certainly, please share with your societies. So if you feel like there's people that aren't represented, we want to make sure. And the first workshop, one of the main questions is who is in here that should be, so that we can sort of target those recruitment of our first batch to bring those people into the conversation.
thank you.
Yep. All right, I think we can move on to whatever's next. I don't know what's on the agenda.
I think that's you, Jen.
Okay, we want to go ahead and talk about field work. All right, I'll do another quick screen share just to inform people of some of the stuff that's going on. So there's two on the sagebrush side of our program. There's a couple of different opportunities. So we'll make this a bit bigger. Let's see if I can make it bigger. So back at the Castle Rocks field site. So if folks are interested in that, this is occurring May 15th through August 4th. Ryan Wickersham, who is Trevor Coughlin's graduate student, along with folks from ISU, will be out. They are going to be doing some drone imagery associated with demographics. So if people want to be part of that, reach out to Ryan. And I don't know if he put his, and you can see that there's a variety of different, both graduate students, undergraduate, and then mid-career. Trevor got tenure, so now he's no longer early career, he's mid-career. So if you're interested in that field work, please reach out to Ryan, and or you can contact me and Peggy, and we can put you in touch with Ryan. The next activity is with Josh at ISU, and so this is an exciting multi-institutional, just like the Castle Rocks is, and so they are going to be out at so let's see, May 15th through August 4th, so similar dates, the Barton Ecological Research Center. Here's some photos of what they're doing. So this looks like a post-burn site, I would guess. And Josh, if you want to kick in anything, you can. Here's some folks planting stuff. So if you want to be part of that, we have Carlos from Boise State, who's going to be doing a fellowship over with Josh and Catherine. And so they're going to be doing stuff over in that area of Idaho. So if you're interested in that, reach out to Josh. And then and again, early career, mid-career, and grad students and undergraduates involved. And then there's two other opportunities. So our Boise State's team, working with Idaho National Labs and the BLM, will be over in the Sand Creek area, National Labs area, leaving April 1st through the 23rd. I know you guys have lots of snow, so we're going to be skiing into our leks to collect things. So if anybody wants to come and ski and collect droppings on leks and plants around the leks, we will direct you where to go, 5 miles one way and back. So it's going to be challenging, I think, this year. And then the last one, and there's our, here's Idaho Falls. The last one is a local one. So this is a, let's see, that's all people going to that. Last one is April 27th and 28th. Anybody who's here in Boise, we have a course associated with the Peregrine Fund this year on restoration. And we had Megan Katu's remote sensing course come out. We've got College of Idaho that had their ecology class come out. And we've been doing collaborative restoration with the Peregrine Fund, the BLM, and a variety of other people at the site. And we were going to be hosting 7th graders again. I think I have a link, but maybe not. So anyway, here's the Birds of Prey, here's the class, and we are going to be hosting 7th graders for 2 days doing rotational stations, including the Idaho Rangeland Research Commission that's going to be out there. I think Kelly might be joining, Gretchen, and then we have CWI, we have Boise State, we have the Peregrine Fund, all doing activities related from soils all the way up to raptors and drones. So if anybody wants to join that 7th grade excitement, come out, and again, that's the last Thursday and Friday of April. And that's what I got for sagebrush related field stuff for the summer. Happy to entertain any questions. And I will drop this link
Thank you so much.
into the chat in case people want to go and read more about it.
Thank you so much, Jen. Chris, over to you.
Great, thanks, Jen. That sounds like a lot of fun. I would love to go ski into some leks, but not going to happen. I've got a less formal summary at ISU. Kyle and Adam are a couple of new grad students in Ernest's lab. They'll be collecting genetic samples for their grad projects. And as we heard earlier, Tyler's working on range-wide phylogenomics of red band trout, and he'll be continuing with that work and hopes to expand the geographic range of some of that work. He and Ernest are at a bioinformatics training workshop along with John Massengale in Virginia right now, which is pretty cool. For some of the common garden work related work, as John talked about, we're going to be wrapping that up in the next couple of months with cohort 3, and then likely be going out in the field to collect a final round from a couple of select populations to work on the thermal preference work that John described. We'll likely be doing that in collaboration with Jake Bledsoe and Mashope to follow up on some of their, or continue the last of their sampling from their seed project looking at trout microbiomes. And then we've got a couple of SARE students who will be continuing to work on several projects, including some trout drift work here at UI and some barcoding work. And I think Jake is going to be hosting a SARE student as well. I think that's the major activities we have planned. I will mention that we've got lots of folks that are planning to go to the Idaho chapter, joint Idaho chapter and Western Division meeting of AFS the first week in May. And so we hope to have a strong showing there. And then there'll be a lot of folks on the trout side at the coastwide genetics meeting later in June, late May.
Great. Thank you very much, Chris. Did you want to add anything with respect to synthesis activities and events as the other item?
Yeah, I'd be happy to. So the other main activity that we'll be working on this spring is synthesis and integration coordination. And hopefully folks saw and responded to the call for registration. That deadline was yesterday. If you email me or Rick, I suspect we'll forgive you and we'll manage to get you on the registration. That is going to be literally hot on the heels of the first use inspired workshop. The first one will be on April 14th from 1 to 4 p.m. Pacific time, 2 to 5 Mountain time. And then the second one will follow on the 18th. And really the goal of those two virtual workshops will be to identify, follow up from the annual meeting to identify specific tangible products that we can produce as part of GEM3 to either synthesize sort of within taxon or within discipline across scales, or what insights we can get from integrating across taxa or disciplines. And some examples of the kinds of products that are being talked about are the secret sauce idea that came out of the annual meeting, where Andrew, Stephanie, Georgia, and Travis are leading an effort to actually survey all of us on what kinds of structural and organizational attributes of GEM3 really supported interdisciplinary work, or perhaps were barriers to interdisciplinary work. And they've got an IRB that they're working on, and they're planning to get the questions finalized during the two virtual workshops in April that will go out to all the GEM3 participants. And those questions are really aimed at understanding those questions in terms of what parts of the organization really helped out, or are there specific attributes of either the researcher or the scholar, whether they were grad student, postdoc, mid-career, late career, how they were supported by GEM3, those kinds of things. The main effort that I'm working on and will be presenting at a couple conferences this spring is a multi-scale synthesis of the trout work and thinking about what insights we can have by kind of zooming out a little bit and thinking about, having done all these studies on a single system, what can we learn about adaptive capacity in changing landscapes? And finally, at the annual meeting, we discussed several potential other projects. One of the ones that seemed to get some traction was to think about trying to get folks together to synthesize what we might learn by comparing the experience across, say, sagebrush and trout. we've done common gardens in both, and we've sort of applied the 3 M's in both groups. And what did we learn within each of those groups, and how did that contrast and compare across those 2 different taxa? And so it really encourage folks to participate at whatever level you can. If your experience is like mine, as I think about like the kinds of papers that really changed my thinking and inspired me to do something really cool, often they were the papers that were review papers or synthetic papers that took a body of work together and brought them together into a more synthetic whole that allowed some insights that weren't possible from single studies. If you can't participate in the virtual or the writing retreat workshop that's planned for July, we still encourage participation through the writing process. And so reach out if you have constraints, and we'll be sure to keep you in the loop on the different projects. Jen, you have anything you want to add?
I've been adding things in the chat to remind people what has gone out in the flurry of emails.
Perfect. Thank you.
Great, thank you so much, Chris. And so just to note that Spencer is not available to present, so we'll find another opportunity to hear on Spencer Root's doctoral research. And so our last item will be an update from Sarah Penny Jackson on the SARE. And with that, I'm going to say thank you very much to everyone and for attending today, particularly to our doctoral candidates for their work and for their presentations, and everyone for being here. And so over to Sarah, who will close up the meeting. Thank you.
Well, I didn't know I was at the very end, so I will be quick. So we have a quick SARE update. Overall, we had 66 student applications and 34 faculty applications. We also, for those that don't know, we increased the payment to $6,000, which really helped a lot. So as of now, we've made awards to about 8 students from 2- and 4-year colleges, and 27 to students at ISU, BSU, and U of I. And of the applicants, we had about 9 URM students from underrepresented populations, and about 42 females who applied. So we're just in the final stages of making awards to students. Award letters have gone out. We still have April 14th as the faculty deadline. So we are still, potentially taking new positions. And then the student deadline is May 5th. And so with that, we just want to, thank the faculty and others for, serving as SARE mentors. It really helps us a lot. And I know a lot of you have taken on more than one student. So we're very thankful for that. And so other than that, that is all I have. And Andy said to go ahead and close out the meeting unless anyone else has anything to add. So thank you.